Thank you, everyone, for joining this presentation. I'm Saeed Izadi, a PhD candidate at Medical Image Analysis Lab, working with Dr. WhiteNNer. And in this presentation, I'm going to talk about the contributions we have had in deep learning-based image restoration with applications on medical imaging. Here's the outline for this presentation. We will start the presentation with going over the thesis context and the applications we target in this thesis. And then we move to a listing of the contributions we made in this thesis. In general, we cover three subtasks of image restoration, specifically image denoising, image super resolution, and attenuation and scatter correction. And we cover two image modalities, two medical image modalities, confocal microscopy and medical imaging. Sorry, confocal microscopy and PET imaging. Okay, let's jump into the thesis context and the applications. Image quality in medical imaging is much different than the generic quality assessment in natural images. And it is a crucial part of many high-risk applications, such as computer-aided diagnosis and interventions, or traffic planning and delivery. With no artifacts, as you see, for example, on the right figure, the clinicians are able to interpret images, medical images, accurately and confidently. However, ignoring the artifacts in medical images may be detrimental to the clinician's interpretation and ultimately disease diagnosis. Colorectal cancer is the second leading cause of cancer-related deaths and third most commonly diagnosed cancer, both in men and women. And we all know the early diagnosis and treatment is crucial for reducing the mortality rate for all types of cancers. We also know that the visualization of these cells forms the major part of screening, monitoring, and treatment of the colorectal cancers. Confocal microscopy, as you see on the right, is a new imaging technology that provides many advantages over standalone histopathological microscopes, as illustrated on the left. For instance, the image acquisition procedure is performed in vivo, which means there is no need for biopsy during the image acquisition. Also, the imaging procedure is less invasive compared to the standalone histopathological microscopes. Since the confocal microscopy imaging is conducted during an endoscopic procedure, the size of the probe has found to be a matter of concerns for the patients and clinicians. And recently, there has been some efforts to miniaturize these imaging devices to reduce the size of the aperture and the cameras. However, this is not an easy task and it comes at some costs. For example, it would give lower resolution images in the outputs because less optical elements fit in smaller devices. Another challenge with confocal microscopy is that the images need to be acquired in low illumination settings to avoid toxic side effects on the living cells and organs. And this explains the reason of most confocal images suffering from the noise artifacts. The classic image restoration solutions based on the image optimization procedure suffer from lack of generalization and expensive computational load at the inference. On the other hand, we have witnessed significant improvements in deep learning for low-level vision tasks in recent years, thanks to access to large data, increased computational power of computers, and development of advanced neural network architectures and optimization procedures. In this thesis, we generally fit the degraded image into the neural network and expect to receive the restored image in the output of the network. So it's going to be a one-to-one mapping between the low-quality image and high-quality outputs. Here's the hierarchy showing our contributions to image restoration literature for medical imaging. During my PhD research, I also published some works on medical image segmentations, which have been excluded from this thesis. For each paper that we discussed throughout these presentations, we categorize the major innovations and contributions across different groups, as you see below: input, architecture, loss, training, and output. Each of these categories basically highlights what type of innovations we are bringing into the proposed method. Okay, in this section, we are going to first focus on the super resolution problem. And this is the first paper that I'm going to present. It was published in MICCAI 2018. And the main novelty in this paper is around the problem definition, problem framing, and investigating how we can use deep learnings to train super resolution algorithms for miniaturized devices. Back in 2017, when we started working on these projects, we noticed that the optics community, they are not using the SR algorithms to mitigate the artifacts in miniaturized devices. However, if they wanted to use super resolution, they had two choices: the classic super resolution algorithms and the deep learning based super resolution algorithms. So given the improvements in deep learning that we observed back in 2017, we decided to pick deep learning and couple it with the other tasks and constraints we have for device miniaturization. So we proposed the first practical efforts to frame the image restoration in miniaturized devices as an image super resolution task. And we accomplished the first feasibility study of leveraging deep networks as the super resolution backbone. In this contribution, we used a densely connected neural network to directly learn the mapping from low resolution images to high resolution counterparts. Here, we visually compare our deep learning based super resolution method against other algorithms. Among the comparisons, we highlight the comparisons against bicubic interpolation, which is being used in many on-device deployments due to its quick and fast runtime execution, and the A+ method, which is the classic example-based super resolution algorithms not using deep learning. Comparison of the red boxes that I highlighted across these three methods reveals that the potential of advanced deep neural network architectures are reconstructing high frequency cues and removing visual artifacts. And numerical results come in this slide, which remark the quantitative improvements of advanced deep learning based networks over baseline and classic super resolution algorithms, highlighted with the red boxes. The next paper we discuss still targets the image super resolution problem. The novelty of this paper, which was published in MICCAI 2019, centers around the architecture design and the attention mechanism. As you can see in microscopic images, as well as in many natural images, many repetitive patterns occur all over the image. And we started to, inspired by the non-local filtering algorithms, we started working on the idea of encouraging the network to take into account the spatial interactions between similar regions during the feature learning. And this would expand the receptive field of the network and result in high quality reconstructions at the end. This figure shows the network we proposed for this task. And as you can see, the backbone of the architecture is a series of residual bilinear attention modules incorporated between shallow feature extractors, sampling layers, and the feature fusion modules. We use a spatial second order attention mechanism to update the feature maps based on the pairwise relationships of different regions. Similar to the previous works in the literature, we leverage the first order statistics as well. Some works like squeeze and excitation networks has already demonstrated the role of channel-wise attention in classification tasks. Therefore, we add the channel-wise attention to our architecture as well. The first order task is just a simple averaging of all pixels in each channel. However, the second order for the second order, we compute an affinity matrix that encodes the pairwise correspondences between all features. We then apply a row-wise convolution to get a 1D attention vector. The same philosophy here for the spatial attention. First order pass is averaging across all channels. And for the second order attention, we first downsample the features, compute the pairwise, apply the 1D convolution, and then scale back the attention matrix to the original full size image. We use overlapping crops from 80% of the CLE 1000 datasets for training. And we also couple the data with augmentation techniques. And we use the rest of the datasets for our evaluations. To ensure that our network works as expected on texture-rich regions, we manually partition the validation set in 2 groups and examine our approach only on the textured images. We compare the performance of our method against the baseline, which is a bicubic interpolation, a classic example-based super resolution, and a similar deep learning-based network which uses the squeeze and excitation attention mechanism, SESR. And you can see that from the top to the bottom, we can see that our network can delicately restore high frequency details such as the granular texture, as you can see in this example or in the middle example, and sudden changes in grayscale pixel intensity. And we highlight the numerical comparisons for 3 methods based on deep learning approaches. The red boxes indicate that our network can improve the final image quality in terms of PSNR across different data sets and scales. Moving to the image denoising section, we start with a study we performed on a large set of existing denoising networks. We investigate all the innovations in existing denoising methods across all categories, such as the input architectures, loss designs, training schemes, and the outputs. This survey has been submitted to the Journal of Artificial Intelligence Review. In this survey, we have studied more than 100 denoising methods and prepared a comprehensive survey which provides a thorough description of the datasets, benchmarks, and metrics, as well as a novel taxonomy of the existing deep denoisers. In the bottom, we have 2 figures. The left one corresponds to the number of peer-reviewed papers published in top-tier conferences for both supervised and self-supervised training schemes. And the right figure shows all the archived and preprint submissions on image denoising. And as you can see over the past few years, the number of papers published on image denoising has been increasing. Also, from the left figure, you can see that the self-supervised denoising has been gaining more interest over the past few years. Until recently, most of the deep denoisers have been leveraging pairs of simulated noisy and clean images for training. In this table here, we show the summary of the synthetic datasets, which are popular in denoising community. For each entry, in addition to the names and the references, we also present some characteristics of the datasets, including the number of images, average resolution, and short information about the semantic contents inside those datasets. In addition to the synthetic datasets, efforts have been made to collect datasets of real, noisy, and clean pairs for training. The table here indicates the summary of the most commonly used real datasets in the denoising literature as well. We studied the existing deep learning-based denoising methods in 2 main groups, supervised and unsupervised, also known as self-supervised groups. We have tables in our survey that summarizes the highlights about the existing works, such as the venue, published year, employed data, architecture, and loss designs. The high-level schematics of the representative works has been sketched and illustrated in the manuscript as well. And we have the same table and high-level schematics for unsupervised and self-supervised denoising methods in the survey as well. The next paper focuses on self-supervised denoising with applications to confocal microscopy images. This paper has been published in Workshop on Visual Recognition and Medical Imaging, ICCD 2019. And in this paper, there are contributions on the architecture loss design, and the output we receive at the end of the network from the network. Collecting noise-free images is infeasible in many applications, and so many unsupervised denoising methods leverage signal priors to restrict the search space to find a clean estimate. Total variation is one of these priors, which encourage the clean estimates to preserve the edges while smoothing out the noise. A few classic image denoising methods have leveraged prior information about the noise for optimization as well. And the motivation of this paper here is to leverage the prior information about the noise statistics in the deep learning networks to further tighten the search space for noise-free image estimation. In this paper, we focus on the white Gaussian noise only, and we propose a neural network for disentangling the signal and noise components in a learned latent space without the need for any ground truth training data. For the inference, we discard the noise branch and only use the denoising branch to get the noise-free estimate. To carefully supervise the training of the network, we leverage 3 sets of losses. Reconstruction loss, which makes sure that the summation of the clean and noise estimates gives the original input. The autocorrelation and stationary loss terms to make sure that firstly, the pixels in the noise image are spatially independent. And secondly, to ensure that the noise statistics are similar across different locations in the image. We also have 2 loss terms for regularizing the clean image, the piecewise constant term and the total variation loss. Here, the highlighted qualitative comparisons show that the WhiteNNer approach can outperform noise to self, while it still lags behind the noise to noise and noise to true, which is basically using the clean ground truth during training. The numerical evaluations indicate that our network can also generate better results compared to noise to self for white Gaussian noise. However, noise to noise and noise to true, or DNCNN, are the winners because they have a stronger supervision during the training. The next paper introduces novel architecture for self-supervised denoising, and it was published in Nikkei 2020. The novelty here is about the architecture we leverage for self-supervised denoising. So the motivation for this paper is, again, to exploit the repetitive non-local patches across different regions to estimate the desired noise-free outputs. To achieve this, we borrow the techniques from the classic patch-based non-local Bayesian filtering algorithm and design a novel patch-based Bayesian network for denoising. Our proposed network has 2 subnetworks, a nearest neighbor network, which finds the most similar patches to a query input patch within the image, and a prior network that takes a set of similar noisy patches and estimates the parameters of the noise-free prior distribution to be used in the Bayesian inference framework. Similar to the previous works, we use the negative log of observational likelihood over the observed noisy patch to supervise the training of the prior network. Given the predicted parameters of the Gaussian distribution approximating the prior distribution, we can compute the parameters of the observational likelihood, again approximated by a Gaussian distribution. And then we can estimate the noise-free patch as in the traditional non-local Bayesian filtering approach using the closed form MAP equation. Here for the visual comparisons, the highlighted red boxes in the last row demonstrate the better quality reconstructions compared to the noise to self. So here, as you can see, compared to the noise to self, which produces over smooth outputs, our network has easier textures across the edges and across this specific region. But still, the noise to true, or DNCNN network, it has a better quality output, which is more similar to the ground truth. This table also summarizes the numerical comparisons between our network, noise to noise and noise to self. Our network consistently outperforms noise to self across all datasets and provides comparable results to the noise to noise. In the last section, I'm going to discuss about the contributions we made for the PET imaging. This paper was presented as an oral paper in annual meeting of Society of Nuclear Medicine and Molecular Imaging, and an extended version of this work has been submitted to medical image analysis. It's under review now. The major contribution about this paper is also around architecture design. In PET images, the subject moves along the axial dimension toward the detector ring, and during the image capturing, the positrons and electrons hit each other, and two photons travel in opposite directions until they reach the detector planes. The line along which the photons travel is called the line of response, and studies reveal that 40 to 50% of the recorded coincidences in a typical whole-body PET scan deviate from the original line of response before reaching the PET detectors, and this would result in loss. Lost photons, which is called attenuation, or inaccurately recorded coincidences, which is called a scatter. Performing attenuation correction based on CT and MR images is very popular in the community. However, they have their own drawbacks. For example, the CT images, they suffer from the artifact propagation drawback, or the CT and PET images need to be registered to have the optimal output. Also, the MR-based methods, the tissue intensities cannot be directly converted into 511 linear attenuation coefficients. There have been some efforts in recent years to directly achieve the corrected PET images from the not corrected PET images. These approaches only operate on the PET images or sinograms without requiring any anatomical information provided by CT or MR. However, in all existing methods that we see in the literature, a single network is used to restore the tracer uptake across all subjects and anatomical regions, while we know that the tracer uptake varies substantially between subjects and between the organs. So to resolve this, we proposed a context-aware network that operates on single 2D slices. So it is very lightweight compared to 3D convolutional networks. However, this architecture incorporates the contextual information from neighboring slices for filtering. The extracted contextual information is used to adjust the convolution kernels in a specific convolution layers, and the input feature is then filtered with the newly updated kernels, which has the information about the context. We develop a customized version of the context-gated convolution for our need and plug it into our architecture. Here are the evaluations. So the first column is the not corrected PET image. The last column is the desired output, which is corrected by the CT information. The third column is the corrected image with UNED, and the last one is the corrected image with our method. Obviously, here you cannot see the differences, but once we look at the quantitative errors, you can see that once we use the context-aware convolutions in our architecture, we can get lower errors for different organs and regions in the body. And here's a profile for the uptake, tracer uptake, showing that with our context-aware convolutions, the estimated uptake tracer uptake would be matching the ground truth much better than the conventional unit architecture. And here's the quantitative evaluations. We calculate the relative error and absolute relative errors, and by plugging in the context-aware convolutions in our architecture, we can bring down the error from 2.43 to 2.11 for the relative error, and 14.79 to 13.96 for the absolute relative error. So in summary, we explored the problem of deep learning-based image restoration to improve the quality of medical images. Our motivation is to advance the state-of-the-art of medical image restorations for the following tasks: image super resolution for confocal microscopy from miniaturized devices. We propose supervised approaches. Image denoising for confocal microscopy. We propose self-supervised approaches and direct attenuation correction for PET imaging. I want to thank my senior supervisor, Dr. Hammerne, for his invaluable advice, continuous support during my PhD, and also my lab mates and colleagues at Medical Image Analysis Lab for the great time we spent together in the lab and in the social settings. And I want to also thank my committee member, Dr. Sava, and all my examiners, Dr. Iguizarian and Dr. Lee, and the defense chair, Dr. Esther, for the time and feedback. Thank you.